you.
Hello.
Yes, Dr. Vikram, you can start.
Yeah, good afternoon, everybody. So today's topic is about biofertilizers, like how you use the biofertilizers. And before that, I just want to give briefly like how you have to isolate these organisms which are used in biofertilizer production or formulation, and similarly how they are mass produced. So to begin with, why we have to use biofertilizer, if you ask me the question, because nowadays you know that due to biomagnification and other things, you are finding the chemicals that are used. It might be the fertilizers, or it might be the pesticides, or uh, fungicides which are used to grow the crops or to control the pathogens or the insect pests that are there. And also a lot of importance has been given for organic agriculture nowadays. And when you say organic agriculture, then like you're not supposed to use any of the chemicals during your crop production. So in this way, biofertilizer has before also it was very very important, but nowadays it's gaining more popularity, and people are trying to move away from these chemicals and pesticides which are used, because in some of the foods and other things, they have found these chemical substances. So, and also some of these chemicals also they have become part of the food chain. So that's why I said biomagnification. So basically, what are biofertilizers? If you ask me, so biofertilizers are basically carrier-based suspensions which are containing live microorganisms, which whenever we apply in to the seed or to the soil, basically it will help in the uh, growth of the plant. In addition to that, it will also help in increased plant yield. In some cases, what happens is it will also improve the immunity of the crop to which you have applied. So whatever organism which you have introduced through the seed or to the soil, what it does is it will basically go to the soil, it will multiply there, it will compete with other microorganisms that are there for the nutrients, space and other things, and then it will survive, and also it will show its reaction on the crop plant. So coming to why we have to use these biofertilizers, if you ask, I would say because of the numerous advantages that we get by use of these biofertilizers. So first one is to begin with, it is not as costly as the inorganic pesticides or fertilizers which we are using. In addition to that, these microorganisms which you introduce, they have the ability to provide nutrients. It might be nitrogen, it might be phosphorus, or it might be K, or it might be other nutrients that are there. They will be made available to the crop plants. In addition to that, these microorganisms which you are using either as biofertilizer or biopesticide, they have the ability to produce different kinds of plant growth promoting substances like indolestic acid, gibberellic acid, cytokinins, and other things. In addition to that, it also improves the fertility of the soil. So it will increase the soil moisture holding capacity, or the water holding capacity, what we call it, will increase the soil aggregation. So overall, the quality of the soil will definitely, it will improve. In addition to that, it is less costly, and also we can easily use. Like I said, it can be treated, it can be given as a seed treatment, or it can be used as a seedling dip, or it can be applied in the main field along with farmyard manure or the compost. So some of these microorganisms which you are using, they might also provide immunity to the crop plant, and also whenever the plant gets immunity, its resistance to plant pathogens and insects which attack them will be very very more. So they will be confident, and they will be withstand the attack from the pests and the pathogens. In addition to that, it is non-toxic to the target organism and also to the plant. It is very very specific to the plant. So we have some biofertilizers like rhizobium, which can be used only for the leguminous crop plants. So coming to isolation of some of these organisms, so the basic procedure for isolation of these organisms is by serial dilution plate count method. We also called as tenfold serial dilution. Here, what we do is we take 10 gram of the soil sample, and then it is mixed with 90 ml of sterile water bank, and then you have to shake it thoroughly so that the soil gets properly mixed with the sterile water. So once that is done, then it becomes 10 to the power of dilution. Then it is serially diluted. You take 1 ml from that, and also it is transferred to 9 ml water blank, which is present in the test tube. That's also under sterile conditions. So once you do that, it becomes 10 to the power of 2. Like that, you keep on diluting until you get about 10 to the power of 5 or 10 to the power of 6. And that dilutions are used for isolation of these different microorganisms. Suppose if I want to isolate phosphate solubilizing microorganisms, then I have to use the media containing that is the Picosia medium I have to use. So this media is also called as tricalcium phosphate medium. And similarly, for isolation of Azotobacter, we use Norris entry medium. And for Rhizobium yeast extract mannitol agar is also used. In this picture, you can see that how serially it is diluted. So the one at the top, you can see that the one which has the cotton with conical flasks. So that is the first dilution. From there, if you transfer to the test tube at the bottom, it becomes 10 to the power of -1. Like that, you go up to about 10 to the power of -6. And from each test tube, you take up 1 ml, and then it has to be placed on the test tube, and later on you are adding the media. So like that you do for all. So in the next picture, you can see that on the left hand side, that procedure is called as pour plate method. On the right hand side, you can see the spread plate method. So in the pour plate method, what we are doing is you are first adding the aliquot or the 1 ml from the dilution to the plate first, whereas in the spread plate, you are adding the media first, and then you are adding the aliquot, and then you are spreading it. Whereas in this case, pour plate method, you are adding the aliquot, and then you are pouring the media to the plate, and then you are slowly turning the plate in clockwise and anti-clockwise direction, so that whatever 1 ml aliquot you have added, it gets equally mixed with the nutrient agar medium. So after that, it is incubated, and then you have to observe for the colonies. So in this picture, you can see that. So this is the picture of phosphate solubilizing organism. We call it as PSM, phosphate solubilizing microorganisms. So here you can see wherever these are the plates containing tricalcium phosphate medium, and you can see that wherever you can see halo zone. So these phosphate solubilizing microorganisms, what they do is they convert the unavailable form of phosphorus into available form. So wherever you can see clear halo zones, that's an indication that the bacteria or fungi has so whatever things that are there, you can observe they are PSBs, phosphate solubilizing bacteria. So they have solubilized TCP, and you can see through the clear halo zone. So in this picture, you can see whatever colonies, pinkish colonies or colonized raised colonies are there. Those are rhizobia. This is yeast extract mannitol agar plate containing Congo red. So the contaminants will take Congo red, and they will be red in color, whereas rhizobium, it is slightly pinkish or colorless in nature, and they are slightly raised. So in this picture, in these plates, you can see that the white and mucoid flat colonies that are there, those are Azotobacter colonies.
Dr. Vikram,
Yes, madam.
sorry to interrupt. Your slides are not changing.
It's not changing now, madam.
No,
So
you're on the first slide.
oh my goodness. Okay, I'll
You have to manually change it. Select the slide which you're talking
now,
on now.
okay, now you can see, madam.
No, still not changed. For
I'm
you, it
doing-
is changed.
Yeah.
1 second.
I mean, okay.
You're on with, yeah, now.
I was on 11. I think I was on 8, 9, 10. Okay,
Yeah.
I'll go again and I'll see your screen sharing. Okay. I'll stop and I'll do again, madam.
Yeah.
Now, madam?
No, still you're on the first slide only. You have to. You can go to on the top toolbar, home insert. From there, you try to change it under slideshow. Home insert designs animations, then slideshow.
Okay, just a second. Actually, where should I go, madam? Here?
On the top menu bar, home insert top toolbar on the left side, on your left. Yeah, just move your
Because I
cursor.
can see, like, in my screen, mute, start video, participant, chat, new
No,
share.
no. On your, I'm talking about your presentation, your PPT.
Yeah, PPT, yeah, slideshow is there.
For me, it's not showing. You can then manually select as you speak.
Yeah, I'm changing, but I don't know.
No, you're on the first slide. Can you move your cursor, bring your cursor to second slide?
Okay. Yeah, I'm in second slide. Yeah,
No, still not changed.
not changed.
Your cursor is not moving. Where is your cursor? That also I can't see.
It's on the right hand side.
For me, it's on the match. Slides on the left, no?
Like,
For me, it is left.
I'm facing the desktop, so it is on
Okay.
my right hand side.
Okay, no,
Now
it's
it's
not changed. You're on the first slide only.
now I came to second slide.
No, like before you in the first sharing when you tried to change it, it changed.
Okay.
So you manually select once again.
Next.
No,
Still
not
not changing,
changed.
madam?
No,
What's happening? I don't understand.
if you have this in, yeah, now it's changed. Now it's changed. You're
Now it's
on
changed.
isolation of bacteria.
Yeah.
Slide.
I think
Fifth slide.
I would go for this one. Then what is that?
Yeah,
Slideshow I would go
now it.
for. Okay. So can you see this?
Yeah. Isolation of PCM. I think you're on 7th or 8th.
Yes, madam.
Yeah, 8th.
So
You're
now,
on 8th. Now it is changed to 9th one.
okay, so I would go for slideshow. If I go for
Yeah.
slideshow, I think it's not moving or like it's not
Yes.
visible.
Yeah.
Okay, so I was on 8th, 9th, and 10th slides.
Yeah.
So in slide number 8, like isolation of phosphate solubilizing bacteria. So here basically you can see PSM is nothing but phosphate solubilizing microorganism. So these are plates, tricalcium plates or plates containing Pekoske agar medium. So here basically these PSMs or PSBs or PSFs, what they do is they convert the unavailable form of phosphorus into available form. So the available form of P taken by the plants are basically H3PO4 and H2PO4. So these organisms convert them into from the organic to inorganic form and make it available to the plant. So in the plate, you can see that wherever clear halo zones are there, the zone clearing is there. So that's an indication that the PSMs are there in it, and also they have solubilized the phosphorus, and that's an indication. So in the next slide, so this is for isolation of rhizobium. So this is a yeast extract mannitol agar plate containing Congo red. So wherever colorless pinkish colonies are there, those ones are rhizobium, and the ones which have taken Congo red color and they are red in color, they are the contaminants. So that's an indication that rhizobium is able to grow. We isolate rhizobium mainly from root nodules of leguminous crop plants. So inside the root nodule, you will have this rhizobium bacteria, and you have to break open the nodules, and then it has to be made into a suspension for the isolation of these organisms. And this is the plate containing Azotobacter, wherein the slimy white mucoid colonies that are there, which are slightly raised in the Petri plate, those are the Azotobacter colonies. So Azotobacter, if I have to give some details about Azotobacter, these are free living aerobic rods of bacteria which have the ability to fix atmospheric nitrogen. And in addition to that, this Azotobacter also has the ability to produce different growth promoting substances like IA or indolestic acid, gibberellic acid, and also cytokinase. And this Azotobacter biofertilizer we can use on different kinds of cereal crops, and even the vegetables, ornamental flower crops, and even fruit and plantation crops. So on these crops you can use, and by using this Azotobacter as a biofertilizer, you can reduce the use of N fertilizers. So nitrogen we are supplying it through ammonium sulfate and also through urea. So by using this, you can reduce the usage of N fertilizers by about 25 to 30%. Similarly, you can also increase the yield by using this biofertilizer by up to about 20 to 25% yield increase can be observed in different crop plants, which I mentioned earlier. Similarly, next is rhizobium. So rhizobium, it has a very good, it is grown on commercially on a larger scale, wherein it can be used on various leguminous crop plants like, cowpea, green gram, red gram, black gram, peas, horse gram, and other leguminous crop plants. So rhizobium is an association, symbiotic association between rhizobium bacteria and also leguminous crop plants. So here rhizobium, what it does is it will provide nitrogen to the crop plant, and this crop plant, what it does is it will protect the bacteria from oxygen damage, because rhizobium contains nitrogenous enzyme which helps in the fixation of nitrogen, and this nitrogen enzyme is highly sensitive to oxygen. So basically it forms root nodule, and also it fixes atmospheric nitrogen. And this rhizobium, you have different rhizobium for different leguminous crop plant. Rhizobium of cowpea cannot infect soybean crop, and rhizobium of cowpea cannot infect red gram. So it is highly specific. So how it is specific means, this rhizobium, it produces polysaccharides, and the roots of plants, different leguminous plants, they produce lectins and flavonoids, and these lectins or flavonoids, they are recognized by the polysaccharides which are produced by rhizobia, and that's how they come near the root zone and they get attached, and then infection and other things happen, and ultimately they form root nodules, and after formation of root nodules, they help in fixation of nitrogen. And if you use these rhizobium biofertilizers in different leguminous croplands, then you can save N fertilizers by up to about 20 to 25%. Similarly, by its usage, an yield increase of up to about 80 to 20% can also be definitely seen in these croplands. And similarly, PSM, phosphate solubilizing microorganism. So different kinds of phosphate solubilizing bacteria are there, different phosphate solubilizing fungi are also there. Like I said earlier, these PSF or PSP, what they do is they convert the organic form of P to inorganic form, and they make it available to the crop plants. And some of the important PSPs that are involved in biofertilizer production are Pseudomonas fluorescens, Xanthomonas, Bacillus megaterium, and Bacillus subtilis. And similarly, some of the bacteria, so some of the fungi which can be used as P solubilizers are Trichoderma hasianum, Penicillium chrysogenum, and Aspergillus niger. And there are different mechanisms by which these phosphate solubilizers, they solubilize P. One such mechanism is by production of different kinds of organic acid. So basically, they produce different kinds of organic acids like gluconic acid, fumaric acid, propionic, acetic, and also formic. And this phosphate solubilizing bacteria or fungi, they can be used on various croplands, almost all the monocots that are there, almost all the dicots, the vegetables also we can use, fruit crops, and some of the commercial crops, and even ornamental flower crops also, it can be used. So by usage, and these P stabilizers also, they have the ability to produce growth promoting substances like IA, gibberellins, and cytokinase. And by usage of this phosphate solubilizers, you can reduce superphosphate application by up to about 25 to 30%. Superphosphate is slightly expensive. So if some amount of P is given through this phosphate solubilizers, it will be very, very helpful to the farmers. And similarly, yield increases of up to about 25 to 30% is also obtained in different croplands by use of different phosphate solubilizing bacteria and fungi, which I mentioned earlier. So we discussed what is biofertilizer and why it is important, what are the advantages we get from usage of biofertilizers, and how these biofertilizers are isolated. So now we have isolated, and the next step is you have to check the efficiency of these biofertilizers, and then they have to be mass produced and under different formulation, and then they can be sold. So what are the steps in mass production of these biofertilizers? So first one is selection of the strain, which is very, very important. Like how you select a seed. If you select a very good seed, then your germination and your crop growth and yield will be better. If the seed only is very bad, then you won't get good yields and good germination. So here also selection of best strain is also very, very important, which releases more, which fixes more N, or which solubilizes more P. That is very, very important. And next one is preparation of primary inoculum. Whatever strain you selected, it needs to be prepared in a proper manner. And then you need the culture broth, the media for this organism to grow. And then inoculation and cultivation of these biofertilizers, and usage of carrier material is also very, very important. Whatever carrier material you use, what it does is it will also help in the adhesion of your culture or the inoculum to the seed, and also it will create a proper environment for the growth of the microorganism that is present within the biofertilizer. And similarly, you have to mix it properly. The carrier material has to be mixed properly with the culture broth or the culture medium. And the next step is curing and maturation and package and storage. So next is selection of strain. I said selection of strain is very, very important, and you have to choose specific strain. Like I mentioned earlier, if it's a legume crop, then obviously whatever strain you have to select is rhizobium. And if it is with respect to nitrogen requirement, then you have to go for either rhizobium or Azotobacter or Azospirillum or algae. And if it is with respect to phosphate P requirement, then you have to go for phosphate solubilizers. So that is also very, very important. And depending on that, you choose whether it is N, P, or other organism. And you have to select a competitive strain. And I mentioned whatever organism which you are adding to the seed or to the soil, that organism has to grow there. It has to compete with other competitors that are there in that particular soil, and it has to withstand that competition. It has to grow, and it has to perform its duty, and it has to survive the environment that is present in that particular space. So it is very, very important there. So in this picture, you can see that I have a pure culture on the left hand side. So from that pure culture, that's called as slant in the test tube. So it's called as slants. From the slant, you take a loopful of the culture, and then you subculture in the Petri plate. So from the Petri plate, you take a loopful culture after it is grown. Then that culture is taken up, and that loopful of culture is inoculated to the primary inoculum media or the culture broth which is there. So it is either 25 ml culture medium, or it is about 50 ml culture medium that is there present within the plate. So from there, you have to transfer that to a big flask containing higher amount of culture medium. It can be grown in shake flask method, or it can also be grown by fermenter method. So fermenter method, you need a large amount of media. So these media which are in the flask, they need to be sterilized separately in autoclave, whereas in the fermenter, within the fermenter only, you can add the culture medium, and then it can be sterilized there itself. So whenever you are adding the culture medium, you have to make sure that the capacity of the culture medium should be about only 50 to 60% of the fermenter or the flask that are there. Why? Because if it is more then during sterilization and other processes, it will overflow and it will get spilled, and contamination chances are very, very high. So once the culture is primary inoculum is ready, once the media has been sterilized, then you have to add the primary inoculum culture to the main culture broth that is there within the fermenter or the flasks that are there, bigger flasks that are there. So whenever you are adding the primary inoculum, the rate is 1:5:10. So suppose you are adding 5 liters of culture broth medium, then you have to add about 500 ml of this 250 to 500 ml of this primary inoculum to that, and then it has to be allowed to run at 120 to 150 rpm. And if it is a fermenter, it will take up to about 1 to 2 days for complete growth of the culture within the medium. So once the culture is ready, specified concentration about 10 to the power of 8 or 10 to the power of 9 ml is reached, then it has to be mixed with the carrier material. So different kinds of carrier material can be used for mixing with the culture medium. So why you are providing the carrier material? So that it will help in the addition of this culture inoculum to the seeds, and also it will provide better environment, which I said earlier. So this carrier material, before using, it might be lignite, it might be talc, it might be peat or vermiculite. It needs to be sterilized properly, and then only it should be used. So whatever carrier material which you use, obviously it should be non-toxic to the organism which you are growing, and it also should have a good moisture absorbing capacity. And whenever you mix it with the inoculum or culture, so it should not form lumps. It should properly get mixed with the inoculum, and it should be easily available in larger quantities, and it should be autoclavable, and it should not be very, very expensive, and also it should have good sticky or addition capacity to the seed, and it should also have good buffering capacity with especially with regard to pH, and it also should not be toxic to the cropland. So basically we can say that it should be inert material. So next step is you mix this culture with a carrier material, and you can also use some of the adhesive agents like carboxymethyl cellulose or CMC. And you can see this in this picture. On the left hand side, it is manually mixed. On the right hand side, you have a mixer or blender, it's called. So in the blender, it is properly mixed, and then you keep it for curing for 1 to a couple of days, and then it has to be added to the polyethylene bags, and the micron size they prefer is about 50 to 75 micron size PP cover, what we call. So that it is mixed, and then it is packed. So once it is packed, the labeling is very, very important, wherein it should contain some of these important information, like name of the culture, whether it is peat solubilizer, whether it is nitrogen fixer, whether it is zinc solubilizer and other things. So all those details are very, very important, and their directions to use should also be given, and the rate of application also should be mentioned, and on which crops it has to be used, that also should be mentioned, and date of manufacture is also very, very important, and date of expiry. So usually for this carrier based biofertilizers, the validity is about 6 months. After 6 months also it will be it can be used, but the population count or the number will be reduced in that. And date of expiry, obviously it needs to be given, and details of the manufacturer and any specific precautions that needs to be taken should also be mentioned on the label. So next, this one is organism. It's called as waste decomposer, which is nothing but a bio-based fertilizer, which contains mainly a group of microorganisms, that is a consortia, which has been isolated from cow dung. So by using this waste decomposer, you can convert the waste material into manure in about 30 to 40 days period of time. So basically, whenever use this waste decomposer, it will improve the fertility status of the soil, and it will also provide immunity to the crop, so to say, resistance to the crop plants to pests and diseases, and also it will help in increase in getting increased crop yields. So this is the picture of a waste decomposer. So in this, like how do you prepare the solution of suspension of waste decomposer is for you need about 2 kg jaggery, and this 2 kg jaggery has to be added um, to 200 liters of water present in a drum, and it needs to be mixed properly. So after mixing it, you have to add 1 bottle of this waste decomposer. One bottle of this waste decomposer will have about 20 to 30 grams of waste decomposer that has to be added to the jaggery solution, 200 liters of jaggery solution, and it has to be mixed properly. So after that, you have to cover the drum with the cardboard or paper, and in a day you have to mix it at least once or 2 times, and in about 5 days period of time, you can see uh, excuse me, creamy growth of the culture. So then it is ready to be used. So in this picture, you can see that jaggery and how they are mixing it to the water, 200 liters of water, and then it is mixed properly, and you can see the creamy solution that is formed, the growth that is formed. So how to use this waste decomposer in converting the waste or compost material is like, you have to first spread on a plastic sheet, you have to spread 1 ton of this compost material, and then you have to spray about 20 liters of the suspension which you have prepared. Waste decomposer suspension has to be sprinkled over this 1 ton compost, and then again you put 1 layer of this 1 ton compost above that, and like that you have to make about 10 layers, and moisture that needs to be maintained is up to about 60%, and you have to mix this thoroughly at 7 days interval, and in about 30 to 40 days period of time, your material will be ready, the manure will be ready, which can be used. So next organism which can be used as a biofertilizer is mycorrhiza, which is a symbiotic association between crop plants and roots of crop plants and fungi. So in this, the main important mycorrhiza are ectomycorrhiza and also endomycorrhiza. Endomycorrhiza, basically we are using it for crop plants, and ecto mainly for forest tree species we use. And to tell you, this mycorrhiza, whatever crops that are there, about 70% of the higher plants are obligately dependent. Obligately means obviously they need the presence of this mycorrhiza in their association for them to grow and perform well. And about 12% are facultatively. Facultatively means it might be needed or it might not be needed, not necessarily needed. And about 18% of them are uninfected. And this endomycorrhiza, it's also called as vesicular arbuscular mycorrhiza, which are obligate symbionts in the sense they need a host for their growth and survival. And these endomycorrhiza or vesicular arbuscular mycorrhiza, mainly they form vesicles and arbuscles in the roots of different crop plants, and these can be used mainly on vegetable crops, even grain and forage crops, commercial fruits, and even in nut and berry plantations, we can use this endomycorrhiza or vascular mycorrhiza. Excuse me. I'm just drinking a bit of water. Sorry.
Sure.
Okay. And some of the organisms belonging to this endomycorrhiza are Glomus fasciculatum, Gigasporum margarita, Acalospora, Sclerosistis. You have Enterospora, Glaziella, and to date about 150 different species of this endomycorrhiza have been identified and they have also been classified. And this you cannot isolate or grow this endomycorrhiza within the lab. Ectomycorrhiza you can grow artificially, but endomycorrhiza it needs a host for its growth. And basically they also produce chlamydospore. And in this picture you can see that the large sac-like structures are called as vesicles, and the thread-like structures are called as arbuscles. So these structures are formed inside the roots of crop plants, and they can be observed by staining under the microscope. So in this picture you can see that whatever plant that is there on the left hand side is the uninoculated plant, and on the right hand side is the picture of the plant which has been inoculated with mycorrhiza. You can see that there is profuse growth of mycorrhiza which can be seen on the right hand side, whereas the growth of root is not very good. And what it does is basically this mycorrhiza, since the growth of the root is profuse, they go deeper in the layer of the soil, and also they get the nutrients and also moisture to the crop. So that's why they are able to provide a good amount of different kinds of nutrients. So that's why it's also called as phosphate mobilizer. And in addition to that, it also helps the crop withstanding drought conditions. It will also provide resistance to different kinds of diseases like caused by fusarium, pythium, and other pathogens. And ectomycorrhiza, like I said, mainly we are using it on forest tree species. And this has a broad host spectrum, in the sense like, one host can be infected by about 40 ectorhizal fungi. One such example is pine tree, which is called as Pinus sylvestris. At a given point of time, it will have 40 ectomycorrhizal fungi infected. So some of the diseases to which this mycorrhiza provides resistance are wilt diseases and root rot diseases, mainly caused by fusarium or even verticillium. Pythium I mentioned earlier, and similarly sclerotium rovsi, and even Phytophthora infestans also gets controlled whenever you inoculate the crop plants with mycorrhiza. And similarly, it also provides resistance to some of these root pathogens by providing mechanical barrier and also chemical barrier. And also these also have the ability to produce growth promoting substances like indolestic acid, cytokinins, and also gibberellins. Now, lastly, coming to the different methods through which you can apply these fertilizers, biofertilizers to crop plants. So first one is it can be applied as seed treatment, and secondly, it can be applied as seedling root drip treatment, and thirdly, it can be applied as soil treatment or application in the main field. And you can also give it in the liquid form through spray application, or it can be added along with the drip irrigation. So in the seed treatment, mainly like it is given as in the dust form, or it is given as a slurry form. So for that you need water, and also adhesive is very important in this case. So adhesives like jaggery or gum arabica or CMC, carboxymethyl cellulose is used. So why we are using is so that this adhesives, jaggery or gum arabica, what they do is they will provide, they will help in addition of this culture to the seed. In addition to that, it will also provide good environment for this microbial culture to grow in that. So if you are selecting jaggery as an adhesive, you need to add about 10% of this jaggery or 40% of gum arabica solution. So once you prepare this mix, then this solution has to be sprinkled. First, before that, you have to take a polythene sheet there you have to spread the seeds to which you have to treat the biofertilizer. So once you spread the seed, this solution has to be sprinkled, and after sprinkling this solution, it needs to be mixed properly. So after that, you have to add the microbial culture, either N fixer or P solubilizer, that needs to be added. It has to be mixed properly, and once it is uniformly mixed, you have to dry it under shade for half an hour, and after that it can be used for sowing, or it's called as dibbling. So you can see that in this picture, they are treating the seed with different biofertilizers. So next method is seedling root dip treatment. So here, mainly what you have to do is you have to take about 1 to 2 kg of biofertilizer, and then it has to be mixed with 5 to 10 liters of water. So mainly this water selection will depend on the amount of seeds that are made. Depending on the seed rate, you select 1 kg or 2 kg, and then this solution you have to dip the roots of the seedlings before transplanting. So you keep the roots dip the roots of the seedlings in this solution of biofertilizer for about 20 to 30 minutes, and then you go for transplanting of those crops. And similarly, if it is paddy, what they do is like you have to make a bed of this size of 2 meter length, 1.5 meter width, and 0.15 meter height. And to this size bed, you have to add about 5 centimeter of water. Height is added, and then you have to add about 2 kgs of this biofertilizer uh, material, and then it has to be mixed thoroughly. And to this mixture or the liquid or solution, you have to dip the roots of this paddy seedling, and it has to be kept for 8 to 12 hours, and later on, after 8 to 12 hours, this can be transplanted in the main field. And here you can see that they have added they have dipped the roots of the seedlings in the biofertilizer solution which has been prepared. And third one is soil treatment application. So here, if you use more than one biofertilizer, you have to mix it separately. So here you have to mix 2 to 4 kgs of this biofertilizer in about 2 to 4 liters of water, and then that particular solution of biofertilizer has to be sprinkled separately on 50 to 100 kg compost heap. So for Azotobacter, you have a separate heap of compost, and for phosphate stabilizer also you have a separate heap of compost. So both are sprinkled separately, and overnight you mix it, and then you allow them to stay as it is. So next day, both can be applied in the main field. And next, this one is use of liquid biofertilizers. So liquid biofertilizers, if you are using it for spray and other purposes, what you can do is you can mix about 5 to 10 ml of the liquid biofertilizer uh, in 1 liter of water, and that can be sprayed to some of the that can be used in drip irrigation also. It can also be used, and some of these can also be used as granules, biofertilizer granules, that can also be applied in the main field. So these are the different methods which can be used for application of biofertilizer. So these are the things which I wanted to uh, tell today. If there are any questions, I'll be very happy to answer them.
Thank you, Dr. Vikram. I'll now take up some questions. Just
Yes,
one
madam.
second. Okay. So the first question is, what career opportunities exist for students interested in working in the biofertilizer industry or promoting sustainable agriculture practices?
Yes, madam. It's a very, very good question. Nowadays, in the beginning of my talk only, I mentioned people are about. I'm not lying. In the past 1 year, I met about 7 to 8 people who are very, very young, and they are working in this one. What is it? Software industry, and they come to us to buy biofertilizer, and they are ready to use and invest this biofertilizer in their crop plants. And once regular people, they come and they do not ask us also, because they have seen the performance of these biofertilizers and biopesticides in their fields. So especially after COVID, people are still more interested in going for organic agriculture. So ample opportunities are there, and companies are also, lots of companies have come up. In addition to that, startup business is also progressing very well. So government is also giving subsidy, like, there was a this one from NABARD. So they give 20 lakhs, and about 8 to 10 lakhs will be given by the NABARD only, and the remaining has to be contributed by the people. So new graduates, some of them, they have ventured into that, and they are doing good business. So here what happens is, biofertilizers, they are also useful in this one. What is that? Nursery, where they are using pro trace. So there also it has the market, and that's why job opportunities also very good, and startup business also can be done.
Okay.
The
And how
awareness
can
among people has increased, madam, especially after COVID. Lot many people have come to agriculture. They want to know, and like in my lab only, we sell about how much? At least 15 tons of biofertilizer and biopesticide formulations we sell. And that's not our full-time job, I'm telling you.
Hmm.
That's
Okay.
not my main job.
Okay.
I'm a teacher,
Yeah.
but still we are doing that.
Okay. That's
I
very
have
good
only
to know.
one person working for that. So we do that. So the awareness in people has increased. Definitely it has increased.
Okay. And next question, how can agriculture students contribute to research projects focused on optimizing biofertilizer efficacy in crop production?
How can students contribute? By doing research.
Yes, research
Okay.
projects.
Yes, definitely. Exactly. I don't remember which this one, DST or DBT. I do not know. There is a this one wherein, like, students can apply for projects through some of the universities, and then they can get projects, and they have to submit their proposal. So they will get funding for 1 year, 2 year, or 3 years. They will get funding, and they can carry out research in this area only. And we can also apply, and the students also can apply for those projects, and they will definitely get funding. If their project is very good, whatever they proposed is of high quality, definitely they will get an opportunity, and they will get funding. And I will check and get back to you, madam, which is that specific section wherein
Okay.
students can apply. Because maybe last year or so I had seen. So definitely they can apply.
Okay.
There are
And
opportunities, and funding is also definitely there.
okay. And how can agriculture entrepreneurs effectively communicate the benefits of biofertilizers to farmers and consumers?
Farmers and consumers. Okay. So every this one, what is that? Every year we do trainings. in our campus also we do, in all other campuses within the university also we do trainings wherein we update them about the things that are there with related to biofertilizers and also other new techniques that have come, and what improvement has happened, what new organism has come. All those things we give the details to the farmers. In addition to that, we also get an opportunity every year. Students who are in final year, they go to the villages and they stay there for about 2.5 months. And during that period of time, they are supposed to work with 5 to 10. Each student has to work with 5 to 10 farmers. So there they also conduct night meetings, and during that period of time, they update them about all these things, not only biofertilizers, all the things that are related to agriculture and horticulture. They do in all the campuses, in all the universities, agriculture and horticulture universities, they do. So we call it as a dissemination of information that we do. It's a sort of extension work, but they are doing. And we also visit the villages during that period of time for the night meetings, and also we also talk about the different aspects, not only biofertilizers, others also. Biofertilizers, definitely we do, the people who are working on that, and others also they do in other campuses and also other universities. That's being done on a regular basis. And we are also they also invite us, the horticulture department and agriculture department people also, 5 to 6 times a year. Definitely they invite us to give a talk on these aspects related to biofertilizers and biopesticides.
Okay. And what are the different types of biofertilizers available, and how can farmers choose the most suitable ones for their specific crops?
Yeah, biofertilizers means, madam, we have mainly N fixers, and already I said N fixers. We have what is that? Rhizobium, specific for leguminous croplands. We have Azotobacter, mainly for cereal and also vegetable crops. Azospirillum, mainly for cereal crops, and for what is that? Sugarcane, we have specific one also. Gluconobacter diazotrophicus, that is specific for sugarcane, beetroot, and other crops. And similarly, phosphate solubilizers, if I say, it can be used on most of the crops. Now we have a waste decomposer. Mycorrhiza can also be used on mainly vegetable crops. We can use, and fruit and plantation crops also it can be used. And you have zinc solubilizers that are available. K solubilizers. So all these can be used in the majority of the crop plants.
Okay.
But only
And
restriction is like rhizobium, it can be used only on leguminous crop plants.
okay, how do biofertilizers contribute to enhancing soil fertility and nutrient availability in agricultural fields?
Okay. Yes. So this whatever organisms which you are using, like I gave the example of waste decomposer. So whenever you add them and these biofertilizers to the soil, what it does is, like, it will improve the fertility status of the soil, and also the soil properties, like, soil aggregation, water holding capacity, and also it will improve the organic matter content of the soil. So when all these things are there, and whatever foliage that falls on the soil, so that will be decomposed, and the organic matter, organic carbon, what we call, so that also will get improved. And these organisms also, like I said, they have the ability to produce plant growth promoting substances. So different kinds of, like, it can be auxins, it can be gibberellins, it can be cytokinins, it can be ethylene. So all these things are produced by them. And these organisms, whenever you're using a consortia, there is one consortia by this one, what is that? Arca microbial consortia from IAHR. So it has nitrogen fixing organism, it has phosphate solubilizer, it has zinc solubilizer, it has K solubilizer. So when you introduce these consortia of organisms, it will provide nitrogen to the plant, it will provide phosphorus to the plant, it will provide K to the plant, it will also provide zinc to the plant. So you are getting major nutrients and also some of the micronutrients also to the crop. So in this way, whenever you're using them, soil fertility, soil aggregation, basically the soil characteristics will improve, water holding capacity will improve, organic carbon content will improve, and these nutrients are also provided to the crop.
Okay. And what are the proper application methods and dosage of biofertilizers for various crops to achieve optimal results?
Madam, like the application of biofertilizer, suppose if it is as a seed treatment, then again it depends on the seed. In some crops, seed rate is high. In some crops, seed rate is very, very less. So if you are using rhizobium for seed treatment, then the application rate is 200 gram per acre, like that. And if it is used liquid biofertilizer, if you are using about 5 to 10 ml per liter, you have to mix. And suppose if it is used as a this one, what is that? In field application, you have to combine with farmyard manure or compost. Then, like I said, for 50 to 100 kg of composted material, you have to add about 2 to 4 kgs in 2 to 4 liters of water, and then it has to be added. So you mix it properly, the solution with the compost, and then next day you have to spread in the main field. So that again depends on the crop. So suppose whatever material you have mixed, the compost with the suspension of biofertilizer, if you are applying for a plantation crop, take the example of coconut, then you have to apply about 16 kgs per plant. To the basin you apply FIM mixed with biofertilizer. So like that, it depends on the crop plant, but general thing is whatever I said earlier. Have I answered that question, madam?
Yes, yeah.
Okay.
Okay. And a few more questions, but we are running short of time. Let me see how soon we can complete it. So,
Yes,
what market opportunities exist for agriculture entrepreneurs in the production and distribution of biofertilizers for various crop segments?
Biofertilizers and biopesticides are working very, very well. But in some cases, what happens is there are people who have large acreage and who know the importance of biofertilizer. They do not care because we have people buying 1 ton, we have people buying 500 kgs, we have people buying 1 kg. So there, what happens? The government also they provide this biofertilizer on subsidy. So there, what happens? People will not buy from us. They will wait for the biofertilizer that is provided on subsidy. So people are using it. So only thing is you have to create awareness, and they should see by themselves that by using this biofertilizer, there is improvement in their crop plant. They are getting better yields, and they want to go for organic agriculture or horticulture. If that comes to their mind, definitely we can improve. There are lots of companies who have come up. There are lots of startups who are working. Some of our students also, they are into this business. They started on a small scale, but they are doing very, very good. So there are good margins also. So like we are producing biofertilizer here. So we are getting 50% of the profit. We are selling at 100 rupees per kg, but still per kg we are getting about 50%, 50 rupees margin. So there is scope for that, for producing, marketing, and also retail. That is, there is scope, definitely.
Okay. And how can universities conduct fields, trials, and experiments to demonstrate the efficacy of biofertilizers in crop yield and soil health improvement?
Yes, madam. We have we have companies who regularly come to us, and also other universities wherein they want to do multi-location trials of their products. So whenever they do, they come, they have to get it done for at least for 3 years. So that is the minimum, this one. So we keep on doing that, and those things also will be shown to the farming community, whenever they visit our campuses for one or the other trainings, we make sure that they see whatever trials and things that are ongoing in our campus. So that's being done in our campus and also other campuses belonging to our university.
Okay. And what research is being conducted on the effectiveness of different biofertilizer strains, biofertilizer strains and formulations for various crop types?
Yeah, currently, madam, we are not doing any research in our campus. We are just mass producing these different kinds of cultures. We have about 10 to 15 different formulations that we have. Those cultures we are mass multiplying, and we are just selling. And whenever we get trials from external sources, we do testing of those biofertilizer materials. Other than that, currently we are not doing any research related to biofertilizers.
Okay.
That's
So,
not in our mandate as of now. So basically, we are not doing right now.
okay. And are there any specific certifications or labels that individuals should look for to identify biofertilizer based products in the market?
Like if I have understood the question properly, like
Yes,
easy
how to
asking, like
identify?
how do I identify whether it is good or bad?
Correct. Yeah.
Good or bad is one thing is like whatever I mentioned, that has to be there in the label. First one is name of the manufacturer, and then what is the active ingredient or the content that is there. So minimum is about if it is a bacteria, about 10 to the power of 8 CFU, colony forming units per gram. That should be there. Per gram is the per gram of carrier material. That should be written there, and all other details related to that should be there, and you have to look for the expiry date. So usually for carrier based, we say 6 months validity, and liquid is 1 year. So after 6 months also, it will be it can be used, but only thing is the population levels will be very, very low. It won't be 10 to the power of 8. It might come down to 10 to the power of 4 or 10 to the power of 5. That is the external thing. Other than that, like if you want to check, then you have to do this colony counts, like I showed. Serial dilution plate count only should be done, and then only you will be able to say whether a specific amount of this one is there or not.
Okay.
Other things,
Now,
by looking at the label only, you have to decide.
okay. So related to the same this thing, another final question as a final question I'll take.
Yes.
What are the potential benefits of consuming crops grown with biofertilizers, and how do they differ from conventionally grown produce?
You mean is it related to the response of the crop?
Uh, it is
No, I mean
consumption.
to say, I mean to say, is he asking whether there is a difference between the crop response to inorganic fertilizers and biofertilizers? That question, what you said?
Nothing as such is mentioned. I'll repeat.
Hmm.
What are the potential benefits of consuming crops grown with biofertilizers, and how do they differ from conventionally grown
Yes.
produce?
Yeah, I got it. So basically, in beginning of my talk, I mentioned about biomagnification. So biomagnification is nothing but within the food chain. In the food chain, like it will start from the producer and it will go to the tertiary, quaternary, like that. The one is at the top. So here in the bottommost organism only, you will have the poison or the chemical that is there which has been applied. So that gets carried over, and it will be at the top of the food chain. So similarly, whatever agrochemicals, insecticides, pesticides which you have applied, most of them they will be within the cropland, and also it will become part of our food. So basically, whatever food you are eating from the grown from using of inorganic chemicals, so it will have more amount of chemicals in that which are poisonous in nature, and chances of people consuming that food getting cancer will also be very, very high. So if you grow crops in an organic way. So these contaminants definitely will not be there in the crop plants, and you will be healthy. Your what is that, life expectancy is definitely going to be high, and chances of you succumbing to cancer also will be very, very less. So healthy food definitely you will get by using biofertilizer compared to the crops which have been sprayed with different agrochemicals or inorganic fertilizers.
Okay. Okay, Dr. Vikram, I don't see any more questions from our participants. So now we have come to end of question round. On behalf of agricultureinformation.com, we'd like to thank you for a very detailed presentation and answering all the questions in depth. And we also like to thank all the participants for joining this meeting. The meeting will now be closed.
Okay, madam. Thanks a lot.
You're most welcome. Thank you.
Bye.